Gentlemen and ladies, welcome to this lecture on transportation of materials into and out of the cell, describing the mechanisms by which cells tend to gain materials for survival and tend to lose materials as well to preserve life. So membrane transport, the discussion is very important in as far as emphasizing the fact that plasma membrane is a boundary that is crucial, and when it is maintained, the cell is able to survive. Why? Because this is the barrier that controls what goes into the cell and what leaves the cell. So the membrane structure results in selective permeability. In other words, it allows some substances to cross easily while others are limited to cross. So this is what we are talking about in terms of structure, using the fluid mosaic theory. We will say that the cell membrane is a bilayer of phospholipids that slide on each other. Embedded in there are proteins that are useful for channels, carriers, as well as those that are associated with glucose units to make cell identity markers. So as mentioned earlier, the cell membrane is selectively permeable. There are a number of mechanisms by which materials cross the cell membrane. Now, based on whether energy is used or not, we can have passive or active transport. Passive transport is where there is no energy that is used, and there are three types of mechanisms under passive transport. We have simple diffusion, osmosis, and facilitated diffusion. For diffusion, it's the solid that is being transported, and osmosis is the solvent, in this case water, being transported. Okay, so in terms of simple diffusion, we consider that the molecules and atoms are in random motion, moving from one side to the other in different directions. But the net movement across the cell membrane is the fact that there will be more movement towards where there is less compared to where there was more. And so what we see in terms of diffusion across the cell membrane is that the membrane has pores large enough that allow molecules to pass through, and because of random movement, some of these pores will end up having some substances crossing through them, and this will happen more often on the side with more molecules. And so if we had molecules that were colored with dye, we'll be able to notice how the colored ones will have moved, yes, in either direction, but mostly in the direction where there was less. Basically, this is what we are illustrating here as well, that where we have two solute substances moving through the solvent and through the barrier, what we'll notice is that the purple color on the right, solutes, will be able to move in all directions, but they will be moving at the end of the day more in the direction of the left. The other colored solute also be able to move in either direction, but the net movement will be in the direction where there was less. So there are factors that will determine whether the membrane will allow some substances to pass through, and this really, the first factor is lipid solubility. The question is the substance able to dissolve in fat? Its partitioning in the fat medium is important. Why? Because a substance that can easily pass through the cell membrane is because it is able to pass through the fat layer of the phospholipid bilayer. And so, highly lipophilic substances will easily pass through the cell membrane. Small molecular weight substances also easily permeate. Charged particles are polar, so they don't easily dissolve in fat. They do not easily pass through the cell membrane. Presence of channels in the cell membrane for particular substances and transporters will determine whether there will be high permeability for that substance or not. So, in other words, we have said hydrophobic molecules which are lipid soluble can pass through rapidly, while polar molecules which are hydrophilic will not easily pass through the cell membrane. But they will need transporter proteins or they will need channels for them to be able to cross. Passive transport processes, therefore, in terms of illustration, what we see is that the highly lipid soluble molecules will need just to cross through the bilayer without needing channels. We have substances like oxygen, carbon dioxide that diffuse readily because of their high lipid partitioning, while substances that are water soluble, they will not easily pass through, like sodium ions, potassium ions, chloride ions. They will need channels, which are sodium channels, chloride channels, calcium channels, and all those. Otherwise, those that are large enough but they can't diffuse in the fat, they will need channels. They will also need the carrier proteins to help them through. Osmosis is the movement of the solvent, in this case water, and what we'll be able to see is that in living systems the solvent is water. Therefore, biologists generally define osmosis as the diffusion of water across the semi-permeable membrane. So in this case, if we're able to illustrate through a U-tube, we separate one side of the U-tube from the other with a selectively permeable membrane that only allows water and not solutes. A solution on either side, we will be able to either attract water because of the high solute concentration. And so over time, we will see that water will have moved into a region where there was more solutes. So because of this, we tend to see that a solution can attract more water based on the fact that they have more solutes. And the ability to attract more water in that environment is called osmotic pressure. Also, there is a term called tonicity, which is the ability of the solution to cause a cell to gain or lose water based on the solute concentrations. So we see that, for example, two solutions with equal solute concentrations will be said to be isotonic, while the one with higher solute concentration will be considered hypertonic compared to the one with lower solute concentration. And the one with lower solute concentration will be said to be hypotonic because it has a higher solvent concentration. And using red blood cells for illustration purposes, we'll be able to see that if we put red blood cells in a hypotonic solution, the red blood cell will gain water and rupture, called lysis. And if we put red blood cells in isotonic solution, the red blood cells will remain without gaining or losing water. But if we put red blood cells in hypertonic solution, the red blood cell will begin to lose water into that solution and it will crenate or reduce in size or shrink. So water balance in cells is very, very important. And so we see for plant cells, they're able to withstand changes in water, but red blood cells do not, or human cells do not, because they do not have a cell wall. So finally, we look at the definition of osmosis. Having looked at osmotic pressure, having looked at osmolarity, really tonicity, we can now define, and this should be your definition, that osmosis is the diffusion of water across a semi-permeable membrane from hypotonic solution to a hypertonic solution. So we end here. Look out for the next discussion of active transport processes.